Hello, today we will be talking about our research project exploring an alternative to antibiotics, T4 phage therapy. Our research seeks to explore the efficacy of the T4 bacteriophage to mitigate bacterial infection of wild-type E. coli and knock out Pgi E. coli mutants. Our research may provide an option to antibiotics typically used to avoid E. coli infection. We chose Pgi because of its essentiality in the glycolysis pathway. In other words, we wanted to know if taking away the Pgi gene would result in a change in the growth of E. coli.
So for the first part of our experiment, we wanted to compare the growth of the parent strain of E. coli where there are no mutations and the Pgi mutant strain of E. coli where the Pgi gene was knocked out and removed, so there may be a lack of glycolysis or metabolism. And this was just to see the significance of Pgi gene and whether it actually affected the growth of E. coli. And for the second and more important part of our experiment, we inoculated the bacteria cultures of the two different strains with the T4 and T4R bacteriophage, which are just viruses that go into bacteria to kill them. And this was to see if these viruses or bacteriophage would be more or less successful in E. coli where this Pgi gene was knocked out. And the way we measured this was through absorbance, which makes sense because cells of bacteria and E. coli are green. And then we also visualize them through a method we use called double agar plates, which basically allows us to see plaques or holes where no bacteria go to seed because of lysis in bacterial lawns. And as a side experiment, we also analyze metabolism, specifically metabolites or molecules related to the Pgi gene in human fibroblast cells infected with HCMV virus. And this was just another way of seeing the significance of the Pgi gene in metabolism.
These three graphs display the results for our research. The first graph displays lysis data of the parent strain of E. coli, which was infected with the T4 phage virus versus the T4R phage virus. This graph shows similar results from both phage strains. The second graph shows similar data to the first, showing minimal change in lysis, but with the Pgi gene knocked out in this strain of E. coli infected with the same two phage strains. The third graph shows metabolite changes when infected with the HCMV virus and shows how certain important molecules for creating energy change in the cell, either increasing or decreasing in level when infected.
In conclusion, the increased lysis among E. coli strains inoculated with the T4R phage is likely explained by the ability of T4R phage to bypass lysis inhibition, thereby increasing infection rates among host cells. However, the significance of Pgi in E. coli metabolism is still uncertain as there is more work to be done. Moving forward, we can reassess the relationship between the knockout strain and T4R phage by focusing on the details of the conditions, such as different media or different temperature ranges. We can also investigate the reason behind metabolite fold change by focusing on environmental conditions and HCMV therapy. Thank you.